In the Meselson stall experiment, they were able to determine that DNA replicated semi-conservatively. In the experiment, they grew E. coli cells first in an N15 medium, then moved them to an N14 medium. If DNA replicated semi-conservatively, we should see an intermediate band between N14 and N15, like in the first picture. And then, after a second round of replication, we would expect to see an intermediate band and an N14 band. Why is this? In the semi-conservative model, the parent strands separate, each being a template strand for the new daughter strand. If we start off with two N15 strands, as I have depicted here in the dark line, each will act as a template for the dotted N14 daughter strand. Here we have two intermediate molecules, and so we would expect to see one peak between N14 and N15. During the second replication, we would expect to see two intermediate molecules and two N14 molecules. This is because the original intermediate strands separated, the N15 becoming another template for a new N14 daughter strand, and the daughter strand N14 separating and becoming the parent strand for a template of a new N14 daughter strand. Thus, we should see one peak at N14, two light DNA molecules, and two intermediates. For the dispersive model, during the first replication, we would expect to see something identical to the semi-conservative model. This is because the theory is that each DNA molecule will contain parts of the daughter's N14 strand and parts of the parent N15 strand. However, we notice that there is something different in the second replication. That is, we still see that there are only intermediate molecules of DNA. This is because, once again, each DNA molecule has parts of the N15 and N14 strand in each. This is notably different in the conservative model. In the conservative model, during the first replication, we would expect to see one N15 molecule and one N14 DNA molecule. This is because the theory is that the original molecule preserves itself. Thus, we should see two peaks, one at N14 and one at N15. During the second replication, we must remember that in the conservative model, the original DNA strand is still conserved. Thus, we should see four new DNA strands, but one is N15 and the other three are completely N14. We should see one large peak at N14 and one small peak at N15. This would be a 3-to-1 ratio.